Good day to Prof. Uyang. We are from Group 25 and we are going to present about the complete genomes of eukaryotic poultry parasite, HISTOMONAS MELEAGRIDIS, linking sequence analysis with virulence or attenuation. Hi, I'm AINA NABILAH A1N6225 and I'm going to talk about the background of this paper. So, HISTOMONAS MELEAGRIDIS is a flagellated extracellular poultry parasite of the order Dichicomonadata. So, it is a protozoan parasite. It causes the disease that is called histomonosis, or called also as histomoniasis or blackhead disease or infectious hepatitis. It is an important disease of gallinaceous birds, especially in turkeys and chickens. For more than 100 years, the introduction of effective prophylactic and chemotherapeutic drugs almost eliminated the disease in the second half of the 20th century. However, there are a ban on the active compound that causes severe reappearance of this disease. The active compound has a nitriimidazole group which has a mutagenic and carcinogenic effects. So in the case, it causes serious sickle lesions and liver necrosis that can lead up to 100% mortality, and in chicken it is less severe, but there are infections that are generally confined to the Zika area. So, preventing mortality, the usage of prototype live vaccines based on an in vitro attenuated strain is shown to be effective but not yet commercially available. Implementation of proper flock management that should be hygienic and early administration of an off-license aminoglycoside antibiotic paramomycins can help to reduce the risk as it contains an anti-protozoal properties. So, this paper has objective of doing molecular studies of phylogenetic positioning, omic-based approach, and in lab doing a propagation through an in vitro xenic culture with turkey or chicken Zika flora and sequencing of genome using monogenic clonal virulent and attenuated strain that are both can be traced to the same single cell. This is to identify the mutation suggested potential virulence targets that were inactivated and modified in course of attenuation.
Hi, I'm Desmond, and I'm going to present the methodology part for this presentation. The first method is to produce protozoan cultures, which consists of two Histomonas MELEAGRIDIS strains from the same single parasite cell and cultured in vitro, xenic to monoxenic transitions with E. coli DH5 alpha. We have 28 passages of virulent and 346 passages attenuated, and it's cultured and maintained in medium 199 with supplements at 41 degrees Celsius. The next is DNA extractions. DNA extracted using QIA amped DNA mini kit. High molecular weight DNA extracted via phenochloroform protocol with ethanol precipitation. DNA quality assessed via qubit, nanodrope, and PEP station. And DNA extraction for variance verification is done by D and easy blood and kit and tissue kit. And for Illumina sequencing, Illumina library will construct by TruSequence DNA PCR free library kit, and Illumina high sequence 3000/4000, which can use pair N 150 base pair reads was used, and the context assembly was done by de novo assemble by the default parameters. And the nanoxpose suite sequencing was done by Oxford Nanopore Minion long read sequencing using SQK LSK 109 1D ligation kit, and flow cells will conduct with 48-hour run and live base scaling. Sequencing run was performed on MinNo software for 48 hours. Base calling enabled bias voltage -180 megavolt. After conducting the Illumina and nanopore sequencing, genome assembly and annotations were then conducted by trimming the adapters using the Pochop and assembled using Fly. Assembly refined with Minimap2 and Raccoon using Illumina reads, and the validations focusing on similarity check via DNA different and completeness check with checkmark. The functional annotations of functional genes made using BLAST2 Go2, and the annotations where the Augustus strand with strand-specific transcriptome repeats regions identified with repeat marker. And the next step is identification and confirmations of variants between strands. SNPs or SNPs predictions on protein stability was done by Provient, aligned genomes using NABME from MABME, and accuracy of variant calling done by Bowtie2, and the PCR conventional and qPCR was both used to confirm deletions and truncations. For the method to characterization of protein bases, where there are a few functional domains identified from the characterizations of proteins, which is kinases, transmembrane proteins, VSP, A motifs, peptidases, and GTPases. And tools used to characterize these functional domains is TMHMM, Fobios, IntelProScan, Provient, Merobes, Kinbase, GTPase spread. And the last method is comparative genomics. The genome of Histomonas MELEAGRIDIS was compared with T. vaginalis, T. foetus, and E. histolytica. And gene family gain or loss inferred using OMA and count with Dolo parsimony. And the functional enrichments was done via Panther. That is the methodology part.
Hi, my name is SHARMIN and we will continue with the results. For genome assembly and annotation, as we can see in table 1, H. MELEAGRIDIS genome sizes are 43 MB, GC content 28%, and approximately 11,000 genes, with little difference between the strains. Introns are rare, with 5.2 until 5.7% of genes, and repetitive regions are approximately 2%. For assembly quality, virulent strain, 99.82% accuracy, meanwhile, attenuated strain is 99.79% accuracy. There are 86% of core eukaryotic genes detected in both strands, and for gene completeness, they are 100% complete CDS in both strands. In figure 1, as we can see, genomes are highly similar for the two strands, except for the 3 inverted contigs in the attenuated strain, and it also has more contigs than the virulent strain. Both strands associated with E. coli DH5 alpha have 99.98% identical genomes. Differences in gene content between the two strains. Gene content of virulent strain are 11,119 genes, meanwhile attenuated are 11,137 genes. 400 genes appeared unique to the attenuated strain, but most were annotation errors, meanwhile 5 were phage contamination. 335 genes appeared unique to the virulent strain, with 234 being annotation errors. Only 2 genes were likely deleted in the attenuated strain. The G7085VIR gene contains a BSPA motif located at positions 38 and 88, suggesting a possible role in virulence. Next result is the confirmation of gene deletions. To confirm the deletions of genes G6116VIR and G7085VIR in the attenuated strain, conventional and real-time PCR were performed on different passage numbers. The deletion of the former gene were verified but could not confirm the deletion of the latter gene experimentally. Further analysis indicated that the former gene deletion happened between passage 83 and 145. Additionally, testing of unrelated Histomonas MELEAGRIDIS strains showed that the former gene remained unchanged throughout in vitro cultivation. Variance identification between strains. After removing the sequencing errors, 68 homozygous SNPs were retained. Of the 68, 17 were located within coding regions. Of these, 4 were synonymous, 12 non-synonymous, and 1 caused a premature stop codon in a protein of the attenuated strain. Out of the remaining 12 non-synonymous SNPs, 2 were deleterious but did not affect functional domains. In terms of indels, an initial set of 21,822 indels was reduced to 2 confirmed homozygous indels. One was a microsatellite insertion in the coding region, which was ignored, while the second was a deletion of a G in the coding region of gene G346, causing a frame shift and a premature stop codon. This frame shift mutation led to the loss of the plex homology domain in the protein and created a short transmembrane domain on the C-terminus, and a profile analysis predicted the mutation as deleterious. Also, G346 was not expressed during in vitro growth, as no transcript was detected. Characterization of protein classes. In Figure A, membrane proteins were predicted using TM-HMM and phobias, hence identifying 1,331 and 1,597 membrane proteins, respectively. Most proteins had a single transmembrane helix. The main types of the membrane proteins are membrane transporters and LRR proteins. A subset of LRR protein is linked to virulence in related parasites. Hence, 94 BSPA motif containing proteins were found, of which 77 had transmembrane domains. Next is peptidase. In Figure B, total of 1,449 peptidases were identified and categorized into 7 families. Metallo and serine peptidases were the most abundant, meanwhile there were also 482 peptidase inhibitors identified. In Figure C, 609 kinases were identified, classified into typical eukaryotic functional classes. Finally, in Figure D, 416 small GTPases were identified, with vast majority belong to the RAP family.
Hi, I will continue the presentation with discussion. First of all is the genome analysis overview. The genome sequencing that has been done using the combination of ONT long reads and Illumina short reads in the assembly of the genome, we found that the genome size of H. MELEAGRIDIS is about 43 megabase, which is surprisingly small compared to its closely related species such as T. virginalis and T. fetus. The reason behind of this genome compactness is because of low of genome duplication levels in the H. MELEAGRIDIS. The overall genome duplication levels in the H. MELEAGRIDIS in both strains is 20% lower compared to the reported duplication in T. virginalis and T. fetus, which around 65% and 61% respectively. Next is the genome comparison between virulent and attenuated strain. First of all is the genome content is similar due to both came from the same parents. However, some of the coding regions show partial support from the AINA rates in the attenuated strain. This indicates that there is a deletion occur in some gene in a certain loci. Reanalysis using PCR reveals that loci deleted is G6116 virulence and not G7085 virulence. The reason behind this is because G7085 virulence has a repetitive nature and similar autologs across the genome, and deletion in the G611 virulence occur in the annotated strain during the in vitro cultivation in the xenic conditions between pasas 83 and 145. The next thing we find out that G6116 gene encodes for the hypothetical protein. Using the blood shears, it reveals that autolog of G6116 in the T. virginalis encode for the putative cell surface adhesive. Thus, we can hypothesize that this gene could be a membrane-associated protein. The evidence to support the hypothesis is that there is a presence of the transmembrane domain towards the C-terminal. Therefore, loss of the potential surface adhesive function in the annotated strain could be a hint to its growth in the virulence of the parasite. Moving on to the analysis of the SNP. A total of 30 homozygous non-synonymous SNP has been detected in the coding region. Among of them is in the gene of G8794/G37, which result in the truncated protein in the annotated strain due to premature stop codon. G37 actually lack a second leucine-rich region domain at C-terminal, which result in the lack of the plex string homology domain, which bind to the phosphoinositidase, and by that targets the protein to the cellular membrane. The mutant protein might lead to the transient interaction with the phosphoinositidase, and this will compromise the phosphoinositidase dependent kinase 1 function for the cells in Lium. However, it is reported that transcript for the G8794 is absent in the transcriptome of in vitro parasite. This indicates that it is a non-constitutive expression, thus contradictory with the PDK1, suggesting it could have a more specific role. The hypothesis, one of the hypotheses is that maybe this gene is expressed in vivo during the infection of the role, and of course, more validation is needed to test this hypothesis. As for the conclusion, based on all the findings that has been discussed in this article, we can summarize it into 3 main points. The first of all is that nanopore long reads and luminous short reads enable us to assemble high-quality genome sequences from 2 phenotypically different strains. The second one is that 2 gene deletions and 2 gene transcription is enough to distinguish between virulence and NA20 strain. Last but not least, the third one is that further experimental investigation is needed to test the hypothesis whether the loss or change of function of these genes might contributing to the annotation. That's all from us. Thank you for listening.